Today, it's a pleasure to have Soojin from biology give our Breakfast Club talk. Everyone may know Soojin. She came here in 2004. Her first two degrees were at Seoul National University in Korea, and then a PhD from the University of Chicago. I didn't realize until I took a look at her CV that she did a little bit of middle school biology teaching back in Korea.
I did.
And she also spent some time, it's unclear from her CV how much, in Scotland. Apparently it wasn't enough to give her a Scottish accent, but...
I could try if I wanted to, but
you
probably don't want to hear it.
How long were you actually there, Soojin?
About a year.
About a year?
Yeah.
Okay. Anyhow, she came in 2004. We're proud to have her as a colleague in our IBB faculty, and Soojin, it's all yours.
Thank you. Thank you. Thank you, Bob, for this opportunity, really. It's really nice. And James and Coley and everybody for organizing this and for wonderful breakfast. It's really nice. Okay. Okay, so I'm going to talk about one of my...
Can you use the microphone just out in the back because it's so loud?
Oh.
Is it on?
It's on for him, I think.
It only records for me.
Right.
Ah, okay. I'm sorry. I thought you had the...
I could try to yell.
Can you hear her in the back?
Can you guys hear me in the back? I tried to yell. Okay. Okay, so I'm excited to talk about one of my main research foci in my lab about DNA methylation and genome evolution. So many of us in the room who may not be in evolutionary biology probably heard about DNA methylation because it is one of the most important types of epigenetic modification. Epigenetic modification means chemically marking DNA or chromatins and so on so that the cell can regulate in addition to the information already in the genetic codes. And DNA methylation is really widespread in all the three domains of life. It's found in bacteria, eukarya, and also in archaea, although it's not clear if the archaea one is originally there or it was laterally transferred. I'm going to talk about one type of DNA methylation, which is cytosine DNA methylation. Okay, as we all know, there's 4 bases, and one of them, cytosine, looks like this. And the enzymes that put methyl groups, DNA methyltransferase, puts the methyl group to the 5 position. So instead of H, now you have CH3, and this is the template. Okay, and this only occurs for C's right next to G in the 3' direction. So these nucleotides are called CpG. So only C and CpG are methylated. And this is the predominant mode of DNA methylation in animal genomes. In plants, there's maybe some other ways. For example, they use C and G. So N means any of the 4 bases. But in animal genomes, it's almost exclusively a CpG, and this makes analysis for us easier than otherwise. So there are many studies going on about the role of DNA methylation, and the emerging theme from these studies is that DNA methylation acts as a silencer or dimmer of gene expression. Okay, the actual mechanism how it does it is still debated, and we'll have a couple of slides describing some mechanisms. But basically, DNA methylation can silence gene expression, and it can also play a role in chromatin compaction. And it can suppress recombination between transposable elements or repetitive sequences. And because of these things, these are related to genome stability. Okay. Also, DNA methylation is the main mechanism how one of the X chromosomes is silenced in females. Also, it plays a role in genomic imprinting. By putting methyl group in C, you can distinguish now between C versus methyl C. So not only you have 4 bases, but you have additional information. And you can distinguish this way 2 alleles from father and mother, and that's how the genomic imprinting works. So these are all functional aspect of DNA methylation that has been widely studied in mammalian models, human and mouse basically. But another important aspect of DNA methylation, which is not always appreciated by molecular biologists, is the fact that it causes mutations and is actually a main source of mutations in animal genomes. And if you cause mutations, it leaves evolutionary signature. So it is a very important source of genome evolution as well. And I'm going to show you what I mean by that. First, let's see how methylation causes mutations. So this is the methylation that I've shown already. So cytosine gets methylated, becomes methylcytosine. And this molecule is chemically unstable. Okay? So it loses the amine group in here very quickly by spontaneous chemical reaction. No enzyme is necessary in this step. So this step occurs very quickly. And then when you lose thymine here and you insert the water molecule so you become oxygen instead of amine, then chemically it's equivalent to thymine. So what they have done, what done is that the cytosine became thymine by methylation and deamination. Okay? So C to T mutation has occurred. And this occurs very frequently. Now, DNA repair mechanism should take care of this, but it's not very efficient because T is naturally found in the genome and it doesn't always distinguish between the T that is mismatched versus T that it should be the real T. So this is, even though DNA repair exists for this mechanism, because this is so rapidly occurring, there's still a lot of mutations that goes through to the next generation that generate from this process. So basically, C to T mutation at CpG sites will create a CpG to TpG, or in the reverse trend, it will be CpG to Cpa. So these type mutations are called as CpG mutations. Okay? And these are really the major source of mutations in animal genomes. I've done some work in my lab to see how often these mutations occur compared to other C to T or G to A mutations. And we have shown that it's about 15 times higher than the normal transitions. And in other animal taxa, there's a wide variety of estimates, but everybody estimates at least an order of magnitude greater than the normal mutations. And in the chimpanzee genome sequencing paper, it was shown that 1/4 of all point mutations between human and chimpanzee has occurred at CpG sites. So you can appreciate how important these mutations are in animal genomes, including our own. Now, one of consequences of this process is that many CpGs will change to TpG. In other words, the CpGs will be eliminated from the genome. So if you look at, for example, human genome, we don't see a lot of CpGs actually because they're all changed to TpG or Cpa at some point and then went through other mutational processes. So DNA methylation will, in effect, reduce the frequency of CpG dinucleotides. And this is an important property that we'll be using in next steps. So in my lab, we have been analyzing the influence of genomic methylation on evolution for a few years now, and we have several directions we have pursued. We have shown that methylation improves genomic molecular clock and also affects compositional evolution. Now, recently we have started looking into the functional aspects of DNA methylation and influence on genome evolution. So these are the two papers that we have submitted and focusing these days. And I'm going to talk about these two today. So let's first talk about methylation and evolution of vertebrate promoters. Now, for the rest of the talk, I'm going to introduce one measure that you need to know, okay? And that is normalized Cpg content, or I say Cpg-OE for simplicity. Cpg-OE is a simple measure that basically is the ratio of observed Cpg in the data normalized by the expected Cpg based upon the random association of C's and G's. So if you have a lot of methylation, then the Cpgs are changed to TpG or Cpa, so this value will decrease, and this Cpg-OE will decrease. So if you have a lot of genomic methylation, Cpg-OE will decrease. And if a gene has been undergone methylation for long evolutionary time scale, the Cpg-OE of that gene will be also lower than genes that has not experienced a lot of DNA methylation. So there's this relationship, and you need to remember this. And this property has been noted early on by, for example, Adrian Bird in 1980. Okay, so if you look at the human genome, then most of CpGs are methylated most of time. Okay, so it is an epigenetic modification. So it can, in principle, change between tissue types and developmental stages, and it does. But for the majority of CpGs, for about 80% of CpGs, for 80% of tissues, they're usually methylated. Okay. Now, because of this, the human genome has generally much fewer CpGs than expected. How few? So this is the distribution of Cpg-OE in the genomic regions. Okay. So if you look at this, then the mean is about 0.2, means that about 20% of expected CpGs are present in the current human genome. And this is basically everywhere. Okay. And this mode of methylation is called global DNA methylation. In other words, in the human genome, if you look at anywhere, it's basically methylated. And Cpg-OE is reduced to 1/5 of what is expected. Now, there's one exception, and very important exception to this rule, and that is the promoter regions. Okay? So promoter regions, some of them are methylated and some of them are not methylated. And indeed, this is what regulates gene expression in case of humans. So generally speaking, if promoters are methylated, then the expression is silenced. And there are many hypotheses how it occurs. Now, one idea is that methyl cytosines can recruit specific proteins for methyl binding domain proteins. And these proteins may themselves inhibit gene expression, or they will recruit other protein complexes that will make the chromatin structure more compact and make it less accessible to transcription factors and so on. So these are some different theories why methylation of promoters will reduce gene expression. But the point is that methylation of promoters can repress transcription. And many promoters are generally methylated, but also there's many that is not methylated. Okay? So genes that is expressed in most of tissues or broadly expressed genes, those promoters are usually not methylated. And it makes sense because you want to have the genes expressed in most tissues, so the promoters are generally hypomethylated. While genes that's expressed in a specific manner in different tissues or different developmental stages, then they tend to be methylated. So there's this distinction of promoters. And this is reflected if you look at the distribution of Cpg-OE in the promoters, then what we see is this very clear by modality. So there's two distributions, one with mean about 0.2, just like the rest of the genome. Okay? But the other one has high Cpg-OE, meaning that there's a lot of CpGs compared to the rest of the genome and close to the expected value of 1. Okay? And if you look at what are these genes, then these correspond to methylated promoters of tissue-specific genes, and these correspond to broadly expressed housekeeping genes. So there's this structural and functional bimodality in the human genome. And I would like to call this curve in the lower CpG part as low LCG, meaning low CpG promoters, and high for HCG for high CpG promoters. Okay? And as I talked about, they are distinct functionally. So high CpG promoters are usually associated with broadly expressed genes, and low CpG promoters are associated with tissue-specific genes. Okay? One more thing that a lot of people get confused sometimes is that now in high CpG promoters, a lot of CpGs are still intact because they are not subject to DNA methylation and mutational decay. So there's many CpGs, many more CpGs than the rest of the genome. And these are often called CpG islands. So it's the HCGs that's associated with CpG islands. Okay? Okay, so even though this promoter bimodality has been noticed before, as I said, most of studies focus on human and mouse models, and there's no idea of how this bimodality evolved and what has caused and when did it evolve? So we started asking these questions. And I thought this is related to the distribution of genomic methylation in animal genomes, which is really not very well known right now. So this is one animal phylogeny. So animal phylogeny is a mess. It's really not resolved yet, but this is one type. So let's just orient ourselves. Humans and mouse belong to the chordates with vertebrates or vertebrate-like structures, and the others are other animals, and this is really very old phylogeny. I mean, the depth of phylogeny is easily greater than 1 billion years. Now, except chordates, the methylation in these other animals is not really well understood, and it was not very well studied either. Now, the reason for that is that two mother species in animals, Drosophila in arthropod and C. elegans in nematode, do not have DNA methylation like in vertebrates. So people thought, okay, maybe methylation don't exist in insects or nematodes and other species, and maybe it's not so important. So it was not studied very much, and even if you look at reviews like published last year or this year, they will say in other animals, methylation is really absent or scarce. And I'm going to show you that it's not true today, okay? But first of all, let's focus on chordates. So chordates are those like vertebrates, and also species like sea squirt, Siona, which has a very primitive vertebrate-like structure. It's not really vertebrates, but it's ancestral chordates. And this is other insects. So distant invertebrates, we don't know the methylation pattern. But within chordates, we have some idea. So the ancestral chordate Siona intestinalis, a sea squirt, many of you not know, but it looks like this. And in Korea, we eat sea squirt. Let me just say that. Maybe not Siona, but some other species. And it's tasty. Okay. Anyway, if you look at sea squirt, the methylation pattern is patchy. And I'll show you what I mean by that. In vertebrates, like humans and other vertebrate species, the methylation is global, and there's other important differences. As we have seen, in humans, methylation is everywhere, does not distinguish between genes and intergenic regions or introns, except some promoters it does distinguish. Now, in Siona, it has been shown that methylation is usually for genes. It's targeted for specific genes. And again, I'll show you what it is. One of the other very interesting differences is that in Siona, transposable elements are not methylated, while in humans, transposable elements are heavily methylated. And actually, because of this observation, some people propose that global methylation occurred because we needed to suppress the genome-wide invasion of transposable elements. So that's one theory, but it's not the case in sea squirt. So if you look at Siona, okay, so I'm going to show you what is patchy methylation. So this is by Adrian Bird's group and published last year early in Genome Research. So this is the gene model, and then they have looked at the percent of methylated CpGs. So if it's 100, that is methylated. If it's 0, it's not methylated. So I've just shown you with the blocks. So it's red is methylated, and blue is not methylated. So let's look at this gene model. So the gene body is methylated, and then if you go intergenic region or promoters, it's not really methylated. Again, if you look at this gene and this gene, you can compare. These two genes are methylated. In between, the intergenic region is not methylated. Now, let's look at these two genes. They are next to each other, but one is methylated, not methylated, and one is methylated. So there's two things that's very obvious. One, methylation is usually for genes, including introns. And two, some genes are not methylated, some genes are methylated. Now, if we looked at Cpg of the genes and also promoters, then it looks like this. So genes are, some of them are methylated, some of them are not methylated. So if you look at the gene, then you have these two curves. The lower one reflecting methylated genes, and the high one reflecting not methylated genes. It's similar to the promoter bimodal in the human, except this case it's happening in the gene bodies. And if you look at the promoter regions, it looks like this. It's around 1, and it's unimodal, meaning that it's not methylated. So again, this reflects the genomic DNA methylation pattern. Okay? And if you remember what you have seen earlier in the talk, this is the complete opposite. of what you see in humans. So I put them together like this. So if you look at a gene model like this, in humans, you have complete methylation of the intragenic regions and these high and low promoters in the promoters, while this is reversed in Sionna. So one of the projects we did is that try to determine when this switch occurred, and also we want to know how this switch happened. So we looked at several vertebrate genomes that were available at that point with good annotation. So we looked at zebrafish, frog, xenopus, chicken, and human, and compared to Sionna, and also we looked at other invertebrates such as echinoderms, sea urchins, which is a little far out over here. What we have seen is that Sionna and sea urchin both look like this, while it is reversed in all the vertebrates. Okay? So the switch has occurred very early on in vertebrate evolution, okay, before the divergence of the fish and other vertebrates. Okay. So let me just point out several things here. So you can see that they look like bimodal, but I learned not to trust my eyes when I do analysis. So we did some statistical analysis to really see if it is bimodal or not. And we used some EM algorithm to basically find the best fit distributions. Is it unimodal or bimodal? And then tested the fits using maximum likelihood ratio test. And it is true that for all the vertebrates, promoters are bimodal and the intragenic regions are unimodal. And another interesting point is that the low one, the LCG, is always similar to this one. So these are in different scales, up to 2 and this is up to 1. But if you look at this, then the lower one is always similar to the introns. So in human, if you look, the low LCG promoters have mean about 0.2, equivalent to the genome-wide mean. And we also decided to look at their functional differences. So in humans, this has been shown that the LCG and HCG promoters correspond to tissue-specific versus broadly expressed genes. So we decided to look at that. And because there's not good data in other species, we decided to look at EST data, which is also a very good indication of tissue specificity. And basically, for all the 4 vertebrate species, the LCG always have lower tissues that they're expressed. In other words, they are more tissue-specific than HCG. And these are all statistically highly significant. So again, in all the species, tissue-specific genes are hypermethylated, and broadly expressed genes are hypomethylated. Okay, so they are functionally distinct. So based upon this, we proposed a model how we can explain that the bimodal promoters have evolved. So we think that what happened is that the promoter originally, ancestrally, was a unimodal distribution with no methylation, so mean around 1. And then the global methylation happened. And global methylation will push this curve toward lower side because of mutational decay of CpG, okay, reduce the CpG OE. And while this is happening, this can be deleterious for genes who has to be expressed in a variety of tissues, because if the promoters are methylated, then the expression is toned down. So natural selection is pushed up for those promoters that are required to be expressed in a variety of tissues. So for broadly expressed housekeeping genes, we think natural selection is preserving the high CpG content. And as a result, the balance between these has created this bimodality. So this is our hypothesis, and we'll see if it's proven true or not. And there are a couple of studies in the literature that looks like supporting our hypothesis. So there's a paper in Nature Genetics 2007 by Weber et al. And what they have done is that they have looked at CpG methylation of human genes, about 20,000 genes, and they have experimentally tested methylation level and CpG OE. And they also compared to the chimpanzee using rhesus macaques as an outgroup to see how often CpG is lost. Now, in their study, they divided promoters to 3 classes, LCG, HCG, and intermediate. But statistically, it's not really significant. But anyway, they did that. But if you just compare LCGs and HCGs, okay, then now this is the loss of CpG. And loss is much higher in LCG, or LCP as they call it, low CpG promoters, than in HCG, while gain of CpG is almost identical in the 3 promoters. In other words, LCGs are losing CpG quickly, more quickly than HCG, like we predicted. So I think it's supporting our hypothesis. Another study was published in MBE. In this case, they have looked at human and mouse promoters using dog as an outgroup. And I have to say this phylogeny is not always true. Sometimes dog goes inside than mouse. But anyway, for this paper, it's not so important. Okay, so when they look at CpG loss and gain of promoters using this phylogeny, what they see is that tissue-specific genes tend to lose CpG, again, as we predicted. So I think so far we have only supports, and hopefully our hypothesis can be proven. So from these observations, we concluded that vertebrate promoters are bimodal, and it is because LCG is formed due to mutational decay of CpGs, which is caused by the global genomic methylation. While HCG promoters have avoided mutational decay by natural selection and thus remained high CpG and formed CpG islands. So that is our current hypothesis. Okay, so I'm going to switch gears and then go to more invertebrates. And this is a paper we just submitted recently and waiting to hear from them. Okay, so let's see. So here I'm going to talk about honeybee genomes. Okay, so let me remind you again that it was understood until really recently, until even now, that many people think that methylation is not very prevalent in animal genomes because the two mother species, C. elegans and Drosophila, do not have DNA methylation. Well, it was a big surprise when they sequenced the honeybee genome and found that all methylation enzymes are there in the honeybee genomes. So if you see all the enzymes, then maybe it's there. So they decided to look at a few genes, and they found that 6 genes, they were able to show experimentally that these genes undergo DNA methylation. So this was a big surprise published in Science in 2006. So this has prompted other people to look at the presence of methylation enzymes in diverse animal genomes. So I'm going to show you that. But before that, let me just remind you, what are the methylation enzymes? So in humans, it looks like this. So there's actually 3 classes or 3 families of DNA methyltransferase, DNMT1, 2, and 3. And we have 3 of the 3. Now, 1 does heritable DNA methylation. So it basically copies DNA methylation information from parents to the children. And DNMT3 does de novo DNA methylation. Now, this guy has only homology to this catalytic domain or C domain, which is highly conserved, and no N domain. But people don't know what does it do, actually. People have no idea what DNMT2 is doing. But it doesn't seem like it's related to heritable or de novo DNA methylation. So it's doing something else, but we don't know what it's doing. But this domain is capable of methyltransferase. So it is included in the DNA methyltransferase. Now, if you look at the distribution of these enzymes in animal genomes, it looks something like this. Okay? So here, mammals, in humans, as we have shown, there's 1 of 1 and 2 and 3 of 3. Okay? And in C. elegans, there's none. In Drosophila, there's only DNMT2, but as I said, because it's not clear what it is doing, people didn't think there was DNA methylation. In fact, if you look at CpG-OE of Drosophila, it's around 1 everywhere. So there's no evolutionary signature of DNA methylation. So these 2 were what people have generally seen and compared to mammals. But now we have more genomes, so we have a fuller picture. Okay, so the honeybee have all 3, and there's 2 of 1, and solitary wasm Nasonia have 3 of DNMT1, and the human head lice that sometimes cause epidemic and make parents go crazy also have all 3. Okay, and recent studies, there was a recent study that showed that actually in C. elegans there's none, but a close relative, P. pacificus, another nematode, has DNMT2. So maybe it got lost here, and maybe it got lost in before somewhere. We don't have any close related genome, so we don't know what's going on here. But if you look at just these insects, it almost looks like perhaps it was lost in here rather than gained in here. So it's not clear, and we have to have more data to really see. But the point here is that just based upon the enzymes, the distribution of them, it looks like methylation may be prevalent in insect genomes. So we decided to look at the honeybee genome in a more detail, see what is going on. So as I said in earlier study, they found these enzymes, and they tried to find what genes are methylated, and they found 6. Now, as I've shown, that we have these methods of Cpg-OE distribution. So we decided to look at that and see if some genes leave evolutionary signatures. So the questions we asked are, what is the pattern of genomic methylation in honeybee? Is it patchy or global, or just a few genes? Okay? And also, we want to see what are the role of DNA methylation. And there were some hypotheses before, so we wanted to check these things. And I'll talk about this in a more detail later. So let's first look at the pattern of genomic DNA methylation. Okay? And we use the distribution of Cpg-OE, again in this case. And this is what we found. Okay? So if you look at Drosophila, again, it's just a unimodal. But if you look at honeybee, you have this clear bimodality again, reminiscent of vertebrate promoters or Sionna genes. And these arrows are those genes that was found in earlier study to be methylated, which are all found in LCG, which we predict to be methylated. Okay? So it looks like a lot of genes are methylated. It's actually roughly half and half. Half belongs to LCG and half belongs to HCG. Okay, so we think that LCGs reflect methylated genes and HCG reflect non-methylated genes. And so far, the genes that we know to be methylated support this distinction. We also analyzed the transposable element. And there's not that many transposable elements that's annotated in honeybee genome, but the marginal element is annotated. So if you look at marginal element, the Cpg-OE is around 1, meaning that it's probably not methylated. Okay? So this actually is very similar to what we have seen in Sionna. So we think that the genomic methylation pattern in honeybee is likely to be patchy. So some genes are methylated and some genes are not. Okay, question?
Question. Was that bimodal distribution shifted to the right? It looked like there were more genes with higher rates.
That's an excellent question, King. I knew you were going to ask that. Yes, absolutely. So if you look at this, the mean of the low curve is about 0.5. The mean of the high is actually about 1.5. So what I think is happening is there's other mechanisms to increase Cpg-OE of HCG. And I think it's related to the fact that these genomes have a really low GC content. So the GC content of these guys is about 20%. So if you just follow, if you just have 20% GC, then you have a really only a few CpG. You expect about 4% of the genome will be in CpG. So to preserve the high CpG content, I think there's a, like, for example, biased gene conversion that's upwardly forcing the Cpg-OE of the HCGs. That's my suspicion. Yeah. Okay. So what is the role of DNA methylation? So to infer this, we first look at if these two proteins in these two categories have different functional annotations. And if you look at this, it's basically gene ontology analysis. And there are many significant enrichment in HCGs and fewer significant enrichment in LCGs. But the pattern really is very clearly different. So genes in LCGs are related to these categories like translation, biosynthesis, and response to DNA damage, while genes in HCGs are all related to cellular communication, signal transduction, and more importantly, system or organ or cellular development. Okay? And these are all highly enriched in HCGs. So we can sort of broadly classify them as housekeeping for LCGs and sort of development specific for HCGs. I'm going to talk about another implication of DNA methylation and its function in honeybee. And this is sort of a really exciting sort of new ideas. So I want to take a small detour to talk about what is so interesting about honeybees. Now, honeybees and our social insects are really interesting because of their phenotypic plasticity. Right? These are like model system for phenotypic plasticity. Now, what I mean by that is that, let me just point to you that, if you compare queen versus worker, these are both female honeybees, and they have the same genomes, but they are so different in terms of phenotype, and not only that, in terms of labor. So workers do work, and queens just reproduce. And they have completely different development. So workers do have only remnant of ovaries and so on, and queen has, of course, full reproductive system. And other aspects of social behavior is really standing out as an extreme case of phenotypic plasticity. And when people first found out about DNA methylation in honeybees, people got excited, and because it's so different from Drosophila, there were some hypotheses that perhaps the methylation related to the phenotypic plasticity in social insects, which is clearly different from the Drosophila. And there was a paper this year that has really shown this may be the case in Science. So in this paper, what the authors did is that they knocked down DMNT3, the de novo DNA methyltransferase. Now, if you do not do the knockdown, then most of the larvae develop into workers, and some of them becomes queen-like. But if you knock down, then most of larvae becomes queens. Okay? And these queens actually had fully developed ovaries and so on. In other words, if you remove DNA methylation, then the honeybee becomes queens, even though it was supposed to be workers. So, in other words, we conjectured that the methylation of queen-specific genes will generate worker phenotypes. Okay? And by reversing that mechanism, we have seen the development of queen-like individuals. And there was another paper that have surveyed how prevalent is genomic DNA methylation in this Hymenoptera. And basically, this shows that there's some appreciable proportion of DNA methylation in Hymenoptera. Many of them are social insects. So this has got to make people in social insects very excited, thinking that perhaps DNA methylation is the mechanism the social insect genomes use to regulate phenotypic plasticity. Because remember, phenotypic plasticity is most likely to be related to gene expression, and methylation is a default mechanism that you can use to really regulate expression of different genes. Okay? So, based upon this idea, we had two predictions. One is that because if you methylate queen-specific genes, you develop worker phenotypes, we proposed that queen-specific genes should be preferentially in HCGs. Now, another way of thinking about this is that if you think there's some genes that can be easily manipulated between different phenotypes, then they should be generally in HCG, because they should not be methylated by default, but methylation of them will turn off those genes and generate specific phenotypes. So we also propose that Cas-specific genes should be also in HCGs. And I would like to really test this hypothesis with more data. So there's one sort of data set that looked at gene expression difference between queen versus worker from adult brain tissues. It's from Grosinger et al. Paper. So we looked at the distribution of ACGs and HCGs for these genes. Now let's go through this diagram together. So the left panel is for LCGs and right panel is for HCGs. Okay? And this is the ratio of queen to worker. So if it's above 0, then it's more expressed in queens. Below 0 is more expressed in workers. It's in the log scale. Okay? And this gray area is what we contributed as sort of no significant difference. Okay? So in this area, genes are basically similarly expressed between queen and workers. And above this gray area is queen-specific, and below this gray area is worker-specific. Okay? So our hypothesis is that the queen-specific genes should be more in the HCG compared to the worker-specific genes distribution. And we can test this by, for example, Fisher's exact test. Okay? And it is sort of marginally significant. So I have to see if it's true or not with more data. Okay. Now the second prediction was that Cas-specific genes should be more prevalent in HCGs, and in this case we got very clear signal that it is definitely more enriched in LCGs. If you look at Cas-specific genes, they look like this, and Cas-genetic genes, they are almost similar or even more in LCG, and which is highly significant. We also looked at literature for genes that was known to be Cas-specific, and we found 28 cases, and 22 of them are all in HCG. So the second prediction seems like it's standing pretty firmly. Okay, so to conclude this part, what we have seen is that the pattern of DNA methylation in honeybee is patchy, and the majority of them are methylated. Half and half are methylated, not methylated. And the role of DNA methylation, at the first look, it does not look like it's silencing transposable elements. But many housekeeping genes are in LCG, so maybe it is to make sure that there's no spurious transcription of housekeeping genes. And also, it is related to Cas differentiation. Specifically, Cas-specific genes are enriched in HCGs compared to LCGs. So these are our conclusions from honeybee genome analysis. We have many other directions that going on in the lab relate to these issues. So we are refining the boundary between the promoter bimodality versus unimodality by looking at species in the boundary of vertebrate-invertebrate evolution. So recently, where the jawless fish lamprey genome is almost completed and it's accessible, so we're looking at that. As we're analyzing Amphioxus, which is another closely related ancestral chordate species to vertebrates. And we plan to analyze other invertebrate genomes using both computational and experimental analysis for the evidence of genomic DNA methylation. And I'm very excited. I have to also say the second part is completely in collaboration with Mike Goodisman, who's sitting here. And also with him, we plan to do more experimental analysis of DNA methylation in social insects. So we'll use two species, honeybee and yellow jackets, right? Okay. And in mammals, at least, we know that level of DNA methylation is directly correlated with gene expression level. And nobody knows if it's the case or not in social insects. So we'll look at this. And also, stemming from our observation, we will tell whether different cats show different levels of DNA methylation. I think that concludes my study. So these are co-authors and collaborators. The most of the work is done by my student Naveen and Brandon Hunt, who's I co-advise with Mike Goodisman. And then people in my lab are all working on aspects of some of these and also some other projects. So I wrote their names, but most of them had to leave for classes. And then these are some fundings. So thank you very much.
Questions?
Yeah?
Is any of these methylase specific for any gene?
Huh? What?
Is any of this DNA methyltransferases specific for a class of gene that might explain what is seen with the honeybee? Because I'm not sure whether just knocking down one gene and you revert the phenotype actually correlate with disease, but in a specific kind of queen-related or population related gene. It may be a global re-repression of genes that will manifest itself in
I see. So you're basically asking if maybe DNMT specifically knocked down the silenced genes that relate to, for example, metabolism.
Exactly.
That's an excellent question. Instead of really it's working to generate phenotypic plasticity. Now to answer this question, we need to know, really all the genes that in HCGs and LCGs in honeybee and see if any of them are different from, for example, Sionna, right? Because the Sionna has this patch methylation. It also has part of gene knockdown, part of gene not methylated and methylated, but they don't have social behavior. So we have looked at it briefly, and what we see is that both Sionna and honeybee have similar genes in LCGs and similar genes in HCGs. So further studies are needed, but my suspicion is that now another way of answering your question is also to see if similar genes are methylated in non-social insect species. Okay, so if you look at insect species that is non-social, yet it has this similar pattern of methylation, and the same genes are knocked down and the same genes are not knocked down, then I think it proves the idea that maybe the evolution of phenotypic plasticity is secondary rather than the first push for the evolution of DNA methylation. Do you agree? So we are looking at this using like Nasonia genomes right now. Yeah. And my suspicion is I'm with you a little bit, but we have to see. Yeah. Oh.
Go ahead.
Okay. Did I understand correctly that the distribution of functions has been switched between vertebrates and the honeybees with respect to the low CG versus high CG?
So that is a very good question. So here now we are talking about the definition of sort of housekeeping. Okay, so it's sort of all started in Adrian Bird's study. So Adrian Bird, when they look at Sionna, they say that LCGs are housekeeping. And when they say housekeeping, that is the genes that is intermediately or lowly expressed but in broad tissues. Now in humans, when we say housekeeping, it's usually genes that's highly expressed in many of the tissues. Now in humans, many of genes that are highly expressed are broadly expressed because these two are expression level and pattern are strongly correlated, as you know. But is it the case in Sionna? Actually, we don't know at all. Yeah. So when I say housekeeping in Sionna or insects, we don't know the expression level, but it looks like a basic cellular processes.
Yeah,
So
I'm
that's
just
sort of
wondering
loose
about
definition.
The possibility of independent evolution of methylation. So the tree
Right.
suggests that maybe it's not the case, but you mentioned we needed more data.
Yeah. Yeah. Yeah.
Soojin, your talk is totally out of my area of expertise, but leads to a lot of thinking on my part.
Okay.
What do we know about the influence of environmental factors on DNA methylation and potentially on evolution?
Yeah, that's a really great question. A lot.
A lot. So
But,
it's a seminar in itself.
No, but that's yeah. I mean, I can tell you a little bit about it. So, for example, these epigenetic signals. The interesting part of it is that it can be it's stimulated by environment. So, you know, we have genetic code we get in the genome, but you can mark some Cs with methyls or other things based upon environmental cues. So, the honeybee experiment, for example, you know, the honeybee queens become queens just because they eat like royal jelly. So different nutrition influences, in this case, hypomethylation. If you don't have a lot of nutrition, you have hypermethylation. You don't express a lot of genes. You don't develop very well. Like in humans, I'm sure it is prevalent because we know that different generations, depending on different nutritions, you grow a lot or you grow less. And it is probably all epigenetic response. So there's a lot. Now, how is it related to evolution is another big question, and I don't think many people really know the answers.
But potentially there isn't it.
I think so. A couple of years ago, there were these studies in plant genomes, for example, that you know, you find this recurrence of this new phenotype that is not in the genome at all. So some people, it was in Nature, and I don't know if there is disproved. I don't think so yet. What people suggested is that, like, for example, we may keep our grandparents' RNA or some chemical signals as a form of RNA somewhere in the genome. So that was the hypothesis in the Nature paper for plants. But what I'm trying to say is that this is completely unknown area, but I think there's these possibilities that some kind of epigenetic signals may be kept in the genome. But again, I am just really speculating here. Okay. Yeah. But I'd like to know, but you know, not. Yeah.
Other questions? We'll make this the last question because there's another group that's supposed to be in here at 9:30. So.
The difference in methylation of transposable elements in
Humans and Sionna?
can it be the difference between DNA elements and Vertebra elements instead of the difference between like vertebrates and non-vertebrates?
That's a good question. So I'm not sure what kind of transposable elements are in invertebrate genomes, but when we looked at, for example, in the human genome, so we can look at DNA elements, Vertebra elements, and separately, then they are all methylated, except their levels of methylation is different. So this could be the possibility, actually. I haven't looked at it. Yeah.
Okay. So, Jim,
Okay.
thank you again very much.
Thank you.